Friends, today we are going to discuss about two main aspects, and those are known as artificial hybridization, that means emasculation and bagging, and second thing will be double fertilization, which is the characteristic of flowering plants. So first of all, we have to discuss about artificial hybridization. What do we mean by artificial hybridization? In this technique, the scientists or the farmers, what they do, they perform fertilization in which the desired pollen is introduced to a desired stigma. And that means what will happen? When the double fertilization or the fertilization takes place, the union of the characters which are desired is achieved. So, in artificial hybridization, it is required to facilitate the hybridization in which the desired pollen and desired stigma are utilized. So, in artificial hybridization, two main aspects are taken into consideration. The first one is known as emasculation, and second one is known as bagging. These are the techniques which are followed in artificial hybridization. So now what happens, friends? If in a single plant, if the plant produces hermaphrodite flowers, that means bisexual flowers, in which carpel is there and stamens are also present, then what will happen? In these kind of flowers, either autogamy or dichomogamy will occur. So, undesired pollen, unwanted pollen will also sit on the stigma, and so that the desirability of the plant will not be achieved. So what happens in emasculation? In emasculation, when the flower itself is in the bud form, that means when it is not open up, at that time with the help of forceps, one has to open the flower, and what happens? The stamens which are there in the bisexual flowers are taken out before the adhesion of the anther. So what will happen? Now, as the stamens are removed with the help of forceps from the flower in the bud form, when the stigma itself is not receptive yet, so the flower from bisexual, from hermaphrodite, it becomes unisexual, that means only female flower. Here no stamen will be there. The stigma itself is not receptive yet. The whole process of removal of stamen from the flower in the bud form, from bisexual flower, is known as emasculation. And now, after emasculation, to protect the flower, when after some time the stigma becomes receptive, to protect the flower from unwanted, from foreign pollen, one has to cover the flower, and that is known as bagging. Bagging means the flower is protected, it is covered by a bag-like structure made up of generally butter paper or copy paper, you can say. And the flower is protected, it is covered. So now what happens? If some unwanted pollen also comes to the flower, it cannot meet the stigma, it cannot reach the stigma. This process is known as bagging. So emasculation and bagging, these are the two techniques from which one can protect the flower from unwanted pollen. Now, after some time, when the maturity of bud occurs, that means bud is converted into flower, when the stigma becomes receptive, at that time, one has, that means the farmer or the collect the desired pollen from the dehisced anther and open the bag, dust the pollen onto the stigma, and again cover the flower with the same bag. So it will protect the other pollens from reaching to the receptive stigma. Now, if someone asks that this is the condition in which the bisexual flowers are present, but if unisexual flowers are present, then so there, if unisexual flowers are present, there is no need of emasculation. No need. Not a requirement. Only bagging technique must be used. In unisexual flowers, the female flowers are covered with the help of bags, and when they become receptive, that means they become mature, the stigma becomes receptive. At that time, pollens are collected from male flowers and they are dusted onto the stigma to achieve pollination and then fertilization. So these are the two techniques which are nowadays utilized in artificial hybridization. Now friends, we have also discussed about pollen-pistil interaction. If the pollen is compatible, if the pollen is self, that means it is desired, it is genetically compatible, then only the pollen will be able to germinate onto the stigma and fertilization will be achieved. We have studied that in the structure of ovule, after the development of female gametophyte, we have studied that 3 antipodal cells are there, 2 polar nuclei comprises of a central cell will be there, and egg apparatus contains 2 synergies and an egg cell. Now, if the pollen is compatible, then what will happen? The pollen tube, after development, after germination of the stigma, will reach to the region of micropyle of the ovule. 2 male gametes which are haploid in nature, they occur in the pollen tubes. We have also studied that the region of synergies towards the micropyle has filiform apparatus which is responsible for the entry of male gametes into female gametophyte during double fertilization event. Sometimes some of the scientists suggest that the filiform apparatus and the synergies produce some kind of enzyme, sometimes cellulase enzyme, which digests the tip part of the pollen tube. And so that what happens at that time, a continuous passage has been created and these male gametes enter into female gametophyte through the cytoplasm of a single synergy. At that time, after some time, the 3 antipodal cells, they become disintegrated and single synergy will also become disintegrated. Now the female gametophyte only has a central cell, an egg cell and 2 male gametes which are released within the female gametophyte. Out of the 2 male gametes which are haploid in nature, 1 male gamete will fuse with the nucleus of egg cell. The egg cell is itself haploid in nature, the male gamete is itself haploid in nature, and so that by the fusion of a male gamete and this egg cell, that is nucleus of the egg cell, diploid zygote is obtained. And this phenomenon is known as syngamy. So this is the first fertilization which occurs in flowering plants, in angiosperms. Now what happens to the second male gamete? The second male gamete will fuse with the secondary nucleus, with the polar nuclei, and it converts them after fusion into triploid primary endosperm nucleus. This is triploid, as I told you earlier, triploid nucleus. So here, two kinds of fusions takes place. First, the fusion involves the egg cell and the male gamete. The second fusion involves the diploid polar nuclei and a male gamete. Now, the central cell after fusion is converted into primary endosperm cell. So here in angiospermic plants, in an ovule, two fusions occur. The first one is known as syngamy. The second one is known as a triple fusion. So two fertilizing events occur in the ovule of angiosperms. And that's why the whole phenomenon, the whole process is known as double fertilization. Now, in future, what will occur? The diploid zygote, which is the result of syngamy, which is located towards the micropyle end of the female gametophyte, it will develop into an embryo. Whereas the primary endosperm cell, which is known as PEC, this is C, triploid primary endosperm nucleus, is known as PEN. The primary endosperm cell is known as PEC. The primary endosperm cell, that is PEC, which is located in the middle of the embryo sac or female gametophyte, will in future develop into an endosperm. An endosperm is nothing but the nutritive structure which provides nutrition to the developing embryo. So friends, here we have studied two things. First one, the process of artificial hybridization with the help of emasculation and bagging. And second, the characteristic of angiosperms, and that is known as double fertilization. Both the theories are important with respect to need as well as good set. Next time we will study about the development of embryo, the development of endosperm in detail.